Meiosis is a process of two successive nuclear divisions, meiosis I and meiosis II. Prior to meiosis, the DNA is duplicated, as it is in mitosis. However, instead of creating two identical daughter cells as is mitosis, meiosis produces 4 gametes that are genetically diverse, and each cell has half the number of chromosomes as the original germ cell. Meiosis produces sex cells such as female egg cells and male sperm cells, while mitosis produces every other cell. When the egg cell and sperm cell meet and make a baby zygote, the new organism being formed will have the normal number of chromosomes, because mama and daddy cell each gave half.
Here you can see a cell getting ready to go through meiosis. The chromatin is condensed in the nucleus, the nuclear envelope is still intact, and the centrosomes filled with centrioles are on one side of the cell. At this point, the nuclear envelope starts to dissolve. Also, the centrosomes start to move to opposite poles of the cell. Here, sister chromatids pair into homologous chromosomes as they start to align at the metaphase plate. Here, homologous pairs bond so tightly that crossing over occurs. This contributes to genetic diversity as different alleles exchange across the chromosomes and will eventually end up in different cells. Here, the centrosomes secrete spindle fibers that will attach to the centromeres of the chromosome. The spindle fibers of one pole attach to the sister chromatid. The spindle fibers from the other pole attach to the corresponding homologous chromosomes. Then the spindles pull the chromosomes apart. Here, cytokinesis occurs. The cleavage furrow forms and splits the cell in two. Each of these new cells has a set of haploid chromosomes and 2 pairs of sister chromatids, which leads us into meiosis II.
Each cell now has a haploid number of chromosomes. No chromosome replication occurs between meiosis I and meiosis II. The nuclear envelope of the 2 haploid cells begin to break down once again as it prepares to enter meiosis II. Spindle fibers form again from the centrioles and the chromosomes begin to move toward the metaphase plate. Meiosis II is similar to mitosis because the sister chromatids line up at the metaphase plate and separate evenly into the daughter cells, just like the chromosomes in mitosis. Because of the crossing over that happened in meiosis I, the 2 sister chromatids are not genetically identical. After cytokinesis occurs once more, 4 genetically unique daughter haploid cells are produced and the nuclear envelopes redevelop. As a result, 8 different possible combinations could be created using the 3 sets of chromosomes. We get this amount by using the 2 to the n power formula, n representing the amount of chromosomes in the haploid cell. In our case, our haploid number is 3, resulting in 8 different combinations.
Non-disjunction can happen in either meiosis I or meiosis II. In meiosis I, the homologous pair of chromosomes are not correctly pulled apart by spindle fibers, which results in 2 haploid cells with more chromosomes than normal and 2 haploid cells with less chromosomes than normal. In meiosis II, the sister chromatids are pulled apart correctly, which results in normal haploid cells, one with more chromosomes than normal and one with less chromosomes than normal. As you can see here, in meiosis I, the homologous pair of chromosomes are incorrectly pulled apart by the spindle fibers, resulting in an abnormal amount of chromosomes in the 4 resulting daughter cells. Consequences of extra chromosomes can result in chromosomal disorders such as Down syndrome.